Hi, and welcome to part 5 of cells. What we're going to look at now is how it is that cells that live sort of side by side stick to each other, stick together, and don't kind of disintegrate one next to the other. You know, how do they glue? How do they glue together, one next to the other in your skin, so that they don't just sort of flop apart? So we have to look and see how that works. And it actually works in 3 different ways. So the cells could form something called tight junctions, or the cells could form desmosomes, or they could form gap junctions. So we're going to talk about those 3 separately. We'll start with the tight junctions. If you had cells like this, you know, this looks like a loaf of bread, but there it is. It's a cell, and then another cell, another cell. This diagram is showing different ways that they can connect. So look up here. Here they're kind of zippering together. Do you notice? One next to the other one. Get my pin out. So they're sort of zippering here and zippering there together. That's one way. Or they could have this type of connection here, which is less of a zipper in one place, and it's got these long strands here that come into the cell. And that looks like this. Or they could actually have these little connectors that allow something to come like this and then out the other side, right? So this is just an anchor, but some of these connectors actually allow the passage of substances, right? So it's more like a little tunnel like this, a tunnel from one to the other that would allow something through. Okay, so we're going to talk about more about them. Zipping together, hooking together, or allowing something to pass directly. So let's look. So first one, tight junctions. It's kind of like a ziplock, right? They glue these cells together pretty tightly. And what happens is that the adjacent cell membranes, they actually fuse a little bit. So we're calling it a partial fusion of those cell membranes. So cell membrane of cell 1 and cell membrane of cell 2 are going to fuse together, and it forms this very tight junction that does not let anything through. It doesn't leak. It doesn't allow anything to pass. So the way that works is that membrane proteins from each cell kind of interlock with each other. So the Ziploc bag is a really good example how, you know, one side of the little plastic locks in with the other one and it seals it shut. So those would be tight junctions. They don't leak. They're not going to let water and solutes through. So if you're thinking of, you know, intestinal cells, you don't want them to leak, right? You just had a big glass of water and you swallow it down your esophagus, and you don't want it to leak out to other parts of your body. You want those cells to be tightly stuck together. All right, and this is an enlarged picture of that, where you have the cell membrane of 1 on this side. So this is cell 1, and this is cell 2 on the other side. And look, this is the membrane of the other one. And right, they are sort of, the proteins here are fusing together and causing them to really attach so that this wouldn't leak. I know it looks like a little hole, but it interlocks very closely. That's a tight junction. Now, how is that different from a desmosome? So this is more like a little patch than a zipper. So it just locks in one place and not in another. It holds the cells together. Yeah, that's good. But leaking would happen, right? If it had fluid in those cells. So it's common in other parts of the body. It's common in places like the epidermis and in the heart. And think of what happens in the epidermis. In skin cells, they get pushed and pulled and twisted in all sorts of directions. So you need to have these connectors between the cells that allow that flexibility, but that are pretty strong. So they resist that stretching and twisting. You can't just, you know, twist your skin off, right? That would be terrible. And the way it does it is has these proteins that hook onto the other cell, and they look like a little J, so they can hook onto the other one. And that makes them very, very strong. So look at this diagram. So again, we have, this would be cell 1 here. And this would be the other cell, the other side, cell 2. And these are intermediate filaments. Remember, this is from the skeleton, the skeleton of the actual cell. And these protein J-hooks here are able to hook on like this from one to the other. And this one's hooking here. If I go to black, it's pretty easier to see. So coming around and coming onto these intermediate filaments, and it's not continuous. It would stop here, but this whole region here then is where that patch is. We're going to think of it as a patch, and that's a desmosome. Now, the third one, remember, gap junction is very different in that it's going to allow something to go through. Up to now we've been trying to secure them together or not allowed fluid. Well, this one does allow fluid. So it allows these small molecules to pass through. It's quite regulated. This is seen in cardiac and smooth muscle, and it's very important in cardiac muscle because gap junctions, these are the intercalated discs that you drew when you had to draw your tissues. Right? You had those little lines that you had to draw in. It's where one cell ends and the other begins. And it's going to allow electrical stimuli to pass directly from one cell over to the next. So it's kind of like a fuse there that's going to allow those ions, and it's going to allow that connection to happen. It also happens in smooth muscle, in our intestine, in certain parts that have their own movement. That have their own peristalsis. I threw a disease in here, celiac disease, which what happens is that there's defective gap junctions. The gap junctions sometimes are too big, and then there's leakiness, and this causes all sorts of problems, digestive problems. So this is what it would look like. Again, cell 1, yellow, cell 2, gray. And you would have these embedded proteins forming this channel in here. It's going to allow the passage from one cell to the other. They could also close. This could rotate and close, and then you don't allow the passage. Very interesting. Okay, so I have a clicker for you now. If you had something like this that was sewing it together in many areas like this, which would you have? Which would you be talking about? Gap junctions, tight junctions, zipper junctions, or desmosomes? Again, the key is going to be at the end of the lecture, and we're going to stop this mini lecture right here. Thanks for joining.